Oh, hey, Caesar.
Oh, hi, April.
You want to hang out today?
I can't. I'm gonna go hang out with my sister right now.
I wish I had a sister.
Yeah, it's fun. They're really there for you. Sorry, but I gotta get going now. Bye.
Man, I wish I had a sister. I wish I had a twin.
Hi, I'm April, and I'm here to describe the basic structure of DNA. DNA stands for deoxyribonucleic acid, and it is in the shape of a double helix, meaning it looks like a twisted ladder. Its two sides are made up of a phosphate and a sugar called deoxyribose, and it goes phosphate, sugar, and this is often called the phosphate-sugar backbone of the DNA. And the two sides run in opposite directions, meaning it's antiparallel, 5' to 3', 3' to 5'. The middle of the DNA is made up of 4 nitrogenous bases, adenine and thymine, which always pair up, and cytosine and guanine, which always pair up. There are two groups of nitrogenous bases, C and T, which make the pyrimidine group, and A and G, which make the purine group. One phosphate, a sugar, and a base pair make a nucleotide. DNA is made up of numerous nucleotides. The different base sequences in the DNA determine the characteristics you have, the physical appearance, and those were passed down to you from generations before you. And you can pass down your genes also to your offspring. This can be done in the process of DNA replication.
Hi, I'm Caesar, and I'm gonna be teaching you about DNA replication. DNA replication is a bunch of proteins that gather together to split the DNA, which is considered a factory. The DNA is fed through these proteins, which will end up splitting into 2 DNA strands.
It's like film through a projector. Okay.
The next protein involved is DNA polymerase, which gets nucleotides and puts them together to make the daughter strand. The next protein that's involved is DNA ligase, which puts the Okazaki fragments together.
Okay, so let's go a little bit more into detail with the replication factory. So we're going to start off with strand separation. And what we have here is the parent strand being split into 2 single strands by helicase. And the point where it's split into 2 single strands is known as the replication port. And keeping these single strands from realigning again, we have the SSBs, or the single-stranded binding proteins. Okay, so now let's look at synthesis. Here we have 2 halves of our original parent DNA strand, and the replication fork right up here. So then we have our DNA strands that are anti-parallel, and one strand can replicate the strand in a continuous motion upwards towards the replication fork, where this is known as the leading strand. However, we have a lagging strand that replicates DNA into short fragments that are later known as Okazaki fragments. And this one actually moves away from the replication fork, making it hard. So whenever replicating DNA, it always starts from an RNA primer. DNA polymers can never synthesize the DNA strand from scratch. It always has to start right off the 3' end of an RNA primer. So here we have our primer, and we see our DNA polymerase moving upwards towards the replication fork away from the primer and starting at the end of the 3'. Okay, so these are our last 2 steps of the DNA replication, only in the lagging strand. Here we have the daughter strand and the RNA primer. We got to get rid of this RNA primer so we can finish synthesizing this strand and then later connect the Okazaki fragments. So we have RNase H that knocks off this primer, and DNase ligase comes in and connects the strand. In DNA, we have the nitrogenous bases, adenine and thymine, that pair together. Yeah. Now we have the nitrogenous bases, guanine and cytosine, that pair together. Together they make Nucleotides with a backbone made of phosphate and sugars, the sugar deoxyribose, and with the nitrogenous bases, thymine and adenine, guanine and cytosine. Replication, DNA replication.
DNA polymerase, which makes the daughter strand. Oh, damn. Stop. I messed up.